Good morning, friends. My name is Manuel Gallastegui, and my group is Santiago Callirgos, Presa, Bernardo Fernandez, and me. And today we're going to talk to you about the cell cycle and more specifically about mitosis. To talk about cell cycle, first we need to talk about eukaryotes and prokaryotes. Eukaryotes have a nucleus and have many organelles, while prokaryotes doesn't have a nucleus but have a nucleoid.
Prokaryotic cells are simpler than eukaryotic cells and have a nucleus with fewer organelles. Binary fission is the basic way of prokaryotic reproduction, and it is as simple as copying and dividing. Eukaryotic cells are more complex and have a more complicated way of cell division.
Eukaryotic cells have a more complex cell division process, which includes mitosis in it. There are two main steps in the eukaryotic cell division process, which is interphase and mitosis. Interphase has three parts, G1, S, and G2.
Interphase. The interphase has three steps. G1, that is when all the cell grows. S, that is when the DNA is duplicated and the two chromosomes stay near the nucleus. And G2, when the microtubules responsible of the division start to generate. Now we are talking about mitosis and all of its steps one by one. Prophase. The prophase is the first step of the mitosis, and this is when the nucleus disappears, the chromatin condenses into chromosomes, the separation of the centrosomes, and the formation of the meiotic spindle. Metaphase. Metaphase is the stage where chromosomes carrying genetic information align in the equator of the cell, before being separated into each of the two daughter cells. The next step is anaphase. In anaphase, the chromosomes that have been aligned thanks to metaphase start separating and going to the opposite poles of the cell.
Telophase is the last stage of mitosis. In telophase, the effects of prophase are reversed. In telophase and cytokinesis, the cell ends its division process and completely separates. Cytokinesis is the part of the cell division process during which the cytoplasm of a single eukaryotic cell divides into two daughter cells. We also want to add that plant cytokinesis differs from animal cytokinesis because of the cell walls. The plants have to grow a double cell wall between plant daughter cells and then separate it. Cytokinesis also largely resembles the prokaryotic process of binary fission, but because of differences between prokaryotic and eukaryotic cell structures and functions, the mechanisms differ.
The first checkpoint appears when the cell passes through G1. Depending on internal and external conditions, it can either delay G1, enter a quiescent state known as GO, or proceed past the restriction point. Following DNA replication in S phase, the cell undergoes a growth phase, known as G2. During this time, necessary mitotic proteins are produced and the cell is once more subjected to regulatory mechanisms to ensure proper status for entry into the proliferative mitotic M phase. Multiple mechanistic checkpoints are involved in this transformation from G2 to M. The mitotic spindle checkpoint occurs at the point in metaphase where all the chromosomes should have aligned at the mitotic plate and be under bipolar tension. When the checkpoints and proteins don't do their control work, cancer may occur. DNA repair processes and cell cycle checkpoints have been intimately linked with cancer. due to their functions regulating genome stability and cell progression. In all, the cell division, which is directly related to the cell cycle, works in different processes depending on the cell. It can be by binary fission. When prokaryotic and eukaryotic cells, the process divides between two main steps, interphase and mitosis. When these processes are not controlled, they can lead to a severe consequence, cancer.